In this video, we'll discuss the biological macromolecules called carbohydrates. Carbohydrates are composed of carbon, hydrogen, and oxygen atoms. They are represented by the following molecular formula. The carbon atoms are linked together, and they're also linked to either hydrogen atoms or hydroxyl groups. The building blocks of carbohydrates are monosaccharides. There are distinct monosaccharides that exist, and because there are so many aspects of their structure that are variable, they form different linkages and they serve different functions as a result. Monosaccharides can have either an aldose or a ketose placement of the carbonyl group. They can have a variation in carbon number. They can have different arrangements of hydroxyl groups in space, and they can also have alternative ring forms. One example of the different arrangements of hydroxyl groups in space would be the sugar molecules glucose and galactose. The hydroxyl group associated with carbon number 4 in the glucose molecule is above the plane of the carbon atom, whereas in galactose, which is also a hexose consisting of 6 carbon atoms, just like glucose, the hydroxyl group attached to carbon number 4 is below the plane of the carbon atom. This confers distinct chemical properties to both these sugar molecules. Sugars can also exist in alternative ring forms, as shown in this image, where the carbonyl group on carbon 1 reacts with the hydroxyl group on carbon 5 to form a ring structure when a proton is transferred to the oxygen atom on carbon 1 and a bond is formed between carbon 1 and oxygen on carbon 5. This leads to two arrangements, one in which the hydroxyl group is present below the plane of the ring, as shown in alpha glucose, or the placement of the hydroxyl group above the plane of the ring structure, as seen in beta glucose. These two are distinct molecules, and as a result can form distinct linkages with other monomeric units. Polysaccharides or complex carbohydrates are polymers of monosaccharide units. The simplest polysaccharides being disaccharides, which are composed of two monosaccharide units. These units can either be identical or different. Monomeric units polymerize to form polysaccharides when a condensation reaction occurs, forming a linkage between two monomeric units called a glycosidic linkage. Examples of glycosidic linkages are shown in this image. In panel A, you have two alpha glucose units reacting with each other via condensation to form a disaccharide called maltose containing an alpha 1,4 glycosidic linkage, meaning that the linkage exists between carbon number 1 of one monomeric unit and carbon number 4 of the second monomeric unit of alpha glucose. Panel B shows a beta glycosidic linkage between a beta galactose molecule and a beta glucose molecule that come together to form a disaccharide called lactose. The resulting linkage is a beta 1,4 glycosidic linkage because its orientation is above the plane of the ring structures. So different kinds of glycosidic linkages can form between these monomeric sugar units to form higher order structures. There are several types of polysaccharides. Some examples include starch, which is a storage polysaccharide in plants. Starch consists of a mixture of branched amylopectin and unbranched amylose alpha-glucose polymers. Another example is glycogen, which animals use as a storage form of carbohydrates. This is a highly branched alpha-glucose polymer. A third example is cellulose, which is a structural polymer found in plant cell walls. Without cellulose, plants would not be able to stand against the force of gravity. Cellulose is made up of beta-glucose monomers. This image shows the diversity in polysaccharide structure. You have starch and glycogen, which are storage polysaccharides in plants and animals respectively. These polysaccharides mainly contain alpha linkages, either alpha-1,6 glycosidic linkages or alpha-1,4 glycosidic linkages, between glucose units, a sugar that is used to derive chemical energy in these organisms. These chains can either be straight or branched. Cellulose is an example of a structural polysaccharide which forms layered sheets that are connected via several hydrogen bonds. This polymer consists mainly of beta-1,4 glycosidic linkages that form between the monomeric units. Structural polysaccharides like cellulose, chitin, and peptidoglycan form long strands with bonds between adjacent strands that may be organized into layered sheets or fibers to give cells and organisms strength and elasticity. Now that we've looked at the structure of carbohydrates, we can start looking at their functions. Carbohydrates serve multiple functions inside the cell. They serve as precursors to larger molecules and provide fibrous structural materials, indicate cell identity, they're involved in cell-to-cell recognition, and they also store most of the chemical energy that is needed by an organism. Structural polysaccharides form fibers that tend to be insoluble, meaning that they exclude water as much as they can. The strong interactions that exist between strands consisting of beta-1,4 glycosidic linkages, in addition to the absence of water within these fibers, makes hydrolyzing these linkages more difficult than alpha-1,4 glycosidic linkages. As a result, the structural polysaccharides are resistant to degradation and decay. This is the reason why it is so difficult to make biofuels out of plant material and biomass. As shown in this image, in most cases, biomass has to be pre-treated with either heat or chemicals to make the cellulose more accessible to enzymes. Another role that carbohydrates play is in cell identity. They help display information on the outer surface of cells in the form of glycoproteins, which are protein molecules embedded in the cell membrane that are linked to carbohydrates via covalent bonds. These glycoprotein molecules help in cell-to-cell recognition and cell-to-cell signaling. As shown in this image, glycoproteins are embedded within the plasma membrane of a cell so that other cells around this particular cell can recognize and interact with it. Carbohydrates perform an important role in storing chemical energy. They're also responsible for providing that chemical energy when cells are in need of it. Most sugars, which are components of carbohydrates, are produced via photosynthesis, which is a key process that transforms the energy of sunlight into the chemical energy of carbon and hydrogen bonds in carbohydrates. When cells need energy, starch and glycogen are hydrolyzed to release glucose units. The alpha-glycosidic linkages in glycogen are hydrolyzed by the enzyme phosphorylase, which is present in most animals, enabling them to break down glycogen and release glucose. The alpha-glycosidic linkages in starch are hydrolyzed by amylase enzymes. Amylase plays a key role in carbohydrate digestion. Amylase is present in human saliva, enabling carbohydrate digestion to begin in the mouth immediately after we eat food. Once enzymes like phosphorylase and amylase break down polymers of glucose to release single glucose units, the energy stored within these glucose units is transferred to a molecule called ATP through the reaction shown on this slide. So essentially, carbohydrate units get oxidized, resulting in the formation of a bond between adenosine diphosphate and a phosphate group, resulting in the synthesis of ATP and the release of carbon dioxide and water. The free energy in ATP is then used to drive other reactions inside the cell that require energy. Carbohydrates contain a large number of CH bonds. These bonds have a high amount of free energy. Oxidizing 1 gram of carbohydrates releases 4 calories of energy. In comparison, fatty acids have even more CH bonds, meaning that they would have more free energy. Oxidizing 1 gram of fatty acids releases 9 calories of energy. So to summarize, when a cell needs energy, reactions lead to the breakdown of glucose. Energy released from this breakdown is then captured through the synthesis of ATP by a third phosphate group being added to ADP, or adenosine diphosphate. ATP makes this chemical energy useful to the cell. That concludes our section on carbohydrates. At the end of this section, I would like you to be able to respond to the questions listed on this slide. Thank you.